To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. So I'm going to continue with the lesson inheritance. You're familiar with the concept of inheritance? Man, animals, plants, all of us inherit characteristics from our parents, from our grandparents, so from the previous generations. So the study about the inheritance process and how this inheritance occurs is what we are discussing under this lesson. So we started off the lesson by discussing common inherited characteristics seen in man. For example, the color of complexion, nature of hair, how the earlobes are, how the thumbs are positioned when you clasp your fingers, or the nature of your thumb. Like that, we discussed some common inherited characters. Then we saw that there are some characters that are not inherited commonly, but rare inherited characters. For example, syndactyl, where the fingers or the toes are fused together. Or polydactyl, where you have an additional finger or an additional thumb, either in one of your hands or legs or both. So then the albinism, the complexion where there is no pigment. So like that, we discuss some rare inherited characters. Thereafter, we understood that some characters, like the development of muscles due to environment, activities, or your food habit, is not inherited by the next generation. So, non-inherited characters. And then we discussed Gregor Mendel, who is the father of genetics. He did a lot of experiments related to inheritance, and for that he used the Pisum sativum plant. Pisum sativum is garden pea plant, and he chose that for various reasons because it had so many good or special features that was useful for his experiment. For example, the short life cycle. It is an annual plant. Then the distinctly differentiable characteristics and the fact that it could be pollinated either by self-pollination or cross-pollination. Again, natural pollination or artificial pollination. So, because of all these characteristics, it was easy for him to use the garden pea plant or the Pisum sativum plant for his experiments. And from the experiments, he explained what inheritance is. Although he did not have the knowledge about DNA, chromosomes, but he was able to explain how inheritance process takes place. So in genetics, once genes are known, knowledge about genes is there, we use symbols. Capital letter to denote the dominant character, simple letter to denote the recessive character. But the same thing was explained by Gregor Mendel. He said there are two factors that determine the characteristic of a particular organism. For example, a tall plant and a short plant. The tall and short are two distinct features, whereas they both refer to the height of the plant. And there are two factors. The factor responsible for tall feature, factor responsible for short feature. But the character is the height of the plant. So, two factors, and out of that, one factor is dominant. So, the factor that is mostly expressed in the case of a heterozygous nature. That is, if there are, we have discussed that before also, when there is a capital T and a simple t, capital T, tall character is expressed. That is because the tall character is dominant over the short character. So, based on that, we were able to explain the occurrence of second generation, F2 generation, and we showed the ratio between different types of plants. You were able to understand that when Mendel did the activity, he used pure breed plants, tall plants, short plants. Then all the first one generation was only tall plants. The short character was not present in the first generation. From that, he was able to understand and explain that the short character is recessive. But in the second generation, when the first generation plants were used as the parents, in the second generation, there was a ratio between tall and short plants. And you were able to understand that the ratio was 3:1. Tall is to short. And when we use the symbols, you saw the ratio between capital T is to simple T, capital T simple T is to simple T. That was 1:2:1. So that is one method of illustrating or explaining the occurrence of the first and the second generation using the monohybrid cross. The same thing can be explained using a method called Punnett square. Now, Punnett square, which was introduced by a scientist called Punnett, can be used to show the occurrence of F2 generation. So, the same thing but showed in a different way. The way it is diagrammatically represented is different. But both of them explain the same procedure. Concept is the same. The illustration is different. So here also, if you consider the same thing, what we did before. So if I go back to the previous slide, I can show you all this is what we did. Now here we consider the monohybrid cross of pea plant, considering tall and short character can be expressed using symbols. So you can remember the parent generation pure breed tall plants and parent generation pure breed short plants. So we took them, we took the gametes, and then we got the first generation. From that we got the gametes and the second generation. So the same thing, the same procedure, we will do it using the Punnett square. So here, for the parent generation, P, parent generation is P. For this, we use squares. So that is why we say Punnett square. So here, the parents are denoted in a square. Capital T, simple t, tall and short characters. So here, just for you all to remember, tall T, short, simple T. Tall capital T means it's a dominant character. Short simple T because it is a recessive character. So those are the parents. From there, we can get the gametes. Now, gametes are usually denoted within a circle. Now, here students, both are TT, capital T is the same factor. So, you don't have to show two gametes. Just showing one gamete is more than enough because all the gametes are going to have the tall character. So, they are gamete. So, if we write the gametes, you get T and T. So, then these two gametes combine and form the first generation, F1 generation. Now, to do that, earlier we directly combined the gametes there. But since we are using the Punnett square, what we do is F1 generation, we need to use the square. So here, of course, you need to have here we will have the gametes, tall character, short character. When the gametes combine, what do you get? Tall and short, you get this combination. All the plants will be tall. So these are the gametes. You show the gamete. You have to have squares, a large square divided into 4 depending on the number of gametes there are. Here both parents produce only one type of gamete. So we need to have one set of squares for one type of gamete, capital T, and here simple T. So then from there, the new generation, F1 generation, is capital T, simple T. All are tall plants. Then from that, we get the next generation. So for that also, we need to have gametes. So here again, we have gametes. Here, of course, we will have 4 types of gametes. How do we have the gametes? Capital T, simple T, capital T, simple T. So earlier in the Punnett square, we had just one for one gamete. Now we will have to have 2 squares. So to get the F2 generation. F2 generation we need to have the gametes and the generation there. So here I will use different colors just to show you all the gametes and the generation. But when you all are doing it later, you can just stick to one color. This is just for the understanding, better understanding. So here T and T, capital T, simple T. Then the other parent, capital T, simple T. Tall character, short character. So we have the gametes in pink color. Then from that, we will get the combination. Now, when these 2 gametes combine, you have to look at the gametes. Tall character, tall character. So from here, this one. From here also, this one. So tall, tall. You can either start from here, tall, tall. Capital T, capital T. Now for this one, here it is the tall character, but here it is the short character. So capital T, simple T. Now for this square, it is from this gamete and this gamete. Although you have the simple T here, by convention, we always write the capital letter first. So you have to use this capital T, then simple T. Then for this square, this T, simple T, and the simple T there. So this will be simple T, simple T. Now from that, can you all understand? Now the black color is the offspring. So that is how you draw the Punnett square. This is not necessary for you all to write when you are writing or illustrating using a Punnett square. This is only for your understanding. We write the gametes here and we write the offspring there. Is that clear, student? And when you combine, you have to look at the squares. Now this square T, capital T, capital T. Here capital T, but this is simple T. So capital T, simple T. Then simple T, capital T, but you have to write the capital letter first. Capital T, simple T. Then these two give rise to simple T, simple T. So that is how you get the plants. And out of these, you all know capital T will be a tall plant. Even though this is capital T and simple T, capital T, that's the tall character, is the dominant character. So that is also going to be a tall plant. Same goes for this one. Capital T, simple T, tall plant. only the simple T combination will give rise to a short plant. So here we will have tall is to short. That is going to be 3:1. 1, 2, 3 tall plants and 1 short plant. Now you can remember the observations of Mendel's experiment. The closest ratio was always 3:1 for the second generation. Is that clear, students? So here tall is to short is this. If we write the ratio between these, capital T, capital T, capital T simple T is to simple T, simple T. What is that ratio? There is 1 here. T, the capital and simple T. There are 2. So 1:2, then simple T, 1. The tall is to short ratio is 3:1 and that is known as phenotype ratio. When we discuss the gene concept, you will understand the character that is expressed is known as the phenotype, and here tall is to short characters. So that way this is known as the phenotype ratio. Whereas this one, this will be later referred to as genes once we study the concept of genetics. So in that capital T is a gene combination. Capital T simple T is another gene combination. Simple T again a gene combination. So based on that, we will call this as the genotype ratio. So phenotype ratio of the second generation is 3:1. Genotype ratio of the second generation is 1:2:1. So these were all from Mendel's experiment. But only thing is, because he did not have the knowledge about DNA, genes, genetics, none of those were known to him. But up to this point, he was able to explain the inheritance, how the first generation occurs and the second generation occurs. So if we quickly go back and try to look at this again. Now Punnett square, which was introduced by a scientist called Punnett, can be used to show the occurrence of F2 generation. I used the same characteristics, tall and short. So for that also, we need to have the parent generation P, both TT for tall pure breed plants and simple T for pure breed short plants. From that we get the gametes, that is this, capital T from here and simple T from there, only one type of gamete each. Then from that we get the F1 generation. We have the capital T, one gamete, the other T, the second gamete. From that the combination T, capital T, simple T. So that is again a tall character. So from here we get the next set of gametes. That also I can show using a line. So from here only we get the gametes from this. So there you can see capital T, simple T, capital T, simple T. When we combine this, when we combine all these, here you can see capital T, simple T, capital T, simple T. I have used different colors just for you all to get the idea. Thereafter, use only one color. When I do other examples, I'll be using only one color. So here we show the gametes in these two boxes, the other two gametes in these two boxes, and the combination of gametes to form the offspring. So capital T, simple T. When capital T and capital T combine, you get that combination. Here capital T, simple T. So capital T, simple T. Again here, although it's simple T and capital T, we always write the capital T first. Then for this one, simple T and simple T here. So that is how you get the second generation, F2 generation. Tall is to short, 3:1. Here capital T is to capital T, simple t, that is 1:2:1. So remember students, using these symbols, capital letter, simple letter, getting these ratios, this is the phenotype ratio, this is the genotype ratio, all that comes after the knowledge of genetics, concept of gene. And based on that, we will be explaining this again. So far what we have done is, we have explained the occurrence of the first generation and the second generation from the Mendel's experiment and the ratio he observed. Thereafter, the Punnett square. I'm sure you all can understand that. So in the next slide also, we will do another example. So to do that, I will go back to the slide where you have the table, you have the information, the readings, the observations of Mendel's experiment. We will look at that and we will select a set of characteristics. So if we go back, this is what Mendel observed. So these are the 7 distinct characteristics that he used, that he obtained or that he observed in his experiment. Color of the flower, purple and white. Color of seed, yellow and green. Shape of the seed, round and wrinkled. Shape of the pod, inflated, constricted. And color of the pod, green and yellow. Position of flower, axial, terminal. And height of the plant, tall, short. We have already used this one. So what I will do is, I will select another one where we use round and wrinkled seed. Shape of the seed, round and wrinkled. So if we take that, now you can see F1 generation is all round seeds. I have explained this to you all. So round is the dominant character. And here also you can see round is in high number. So the ratio again is 3:1. Round is the dominant character and wrinkled is the recessive character. So how do we use the letters? Capital R for round seeds and simple R for wrinkled seeds. So I'm sure you all can do it on your own also. So I would prefer if you do that first and then you can again look at how I'm giving the explanation. So I'll go to the slide there. Right. So here we will use a set of characters, shape of seed, round dominant character. So we use the letter capital R, and wrinkled recessive character. So we use the letter simple R. Using that, we will use the symbols to explain the formation of generations. So we have the parents here, parents or P generation. So parents are written as P generation if they are pure breed plants. Normally Mendel used pure breed plants. So round, capital R is to wrinkle, simple R, simple R. Then what do we have? We have the gametes. So from here we get the gamete R. Here we have the gamete simple R. You can write it in lines, or from there you can use lines like this to indicate the formation of gametes also. Then we have the F1 generation. So here F1 generation. So to get the F1 generation, not much of a complication, by capital R and simple R. So you get capital R, simple R. All round seeds. So as I introduce the terms phenotype and genotype, what is the phenotype? All are round seeds. What is the genotype? Gene combination, capital R, simple R, that is the genotype. There is only one expressed character. Gene expression is only 1. Then, if we get the genes for the second generation. Now, you can remember in Mendel's experiment, he had the parent generation. He made them to undergo cross-pollination by depositing the pollens of round seed plants on the stigma of wrinkle seed plants and the pollens of the wrinkle seed plants on the stigma of the round seeds plant. So that is how cross-pollination was carried out. Then when he used parent generation, he collected the pollens and then he allowed them to undergo self-pollination. So from that only the F2 generation. But we need to have the gametes. Now gametes here you have R and R. So there are going to be 2 parents, both with capital R, simple R. So both of them will form 2 types of gametes. Capital R, simple R, capital R, simple R. Both parents. So then what will happen? From these gametes, the second generation will be produced. F2 generation. Now F2 generation, this will combine with this. What is that going to be? Capital R, capital R. Then this one will combine with the next one. Now you never combine this and this. These 2 gametes are not combined because they are from the same flower, same plant. Here when we combine the gametes, students, you have to remember these 2 were obtained from one set. So we don't combine this capital R with this simple R again. This is one set, this is another set. So always combine one from the first set with the another one from the second set. So we have combined both with the first one. R, capital R and simple R. Capital. This is capital R and simple R. Then we have this one with this one. Again simple R, capital R, but when you write it as capital R, simple R. And finally, these 2 combine, giving rise to simple R, simple R. Now you can see all 3, capital R, capital R, capital R, simple R, capital R, simple. All of them, since they have the round seed character and that is the dominant character, all of them will have round seed. So all 3 round. And this will be wrinkled. So like I said, if we take the ratio. Now remember these students, the ratio F1 generation what? All round seeds. And the factors combined, all capital R, simple R. Then if you take the F2 generation, we have the ratio. What is it? We have round is to wrinkled, that is equal to 3:1. That is one ratio. What we'll be calling this as phenotype ratio. Then capital R is to capital R, simple R, simple R, simple R. This is going to be 1:2:1. So can you all understand this now? Whatever the characteristic you choose from the Mendel's experiment, you get the same ratio. Always F1 generation, the dominant character is expressed. the recessive character is not expressed. So all are round seeds. In the second generation, dominant is to recessive character, that is round is to wrinkled as 3:1. And the ratio between this combination, factors RR, capital R, capital R, capital R, simple R is to simple R, simple R, that is always 1:2:1. That is how we show the occurrence of the first and second generation. Is that clear to you? So the same thing, students. Now we can do it using the Punnett square. I will show you all that as well. So here I am sure you all have understood the occurrence very clearly. Parent generation, gametes, F1 generation, again gametes, and finally the F2 generation. So after this we will use the Punnett square method. So here we will use the second method. Punnett square. So already I have written the characteristics, so I am not going to repeat that again. If we write the parents, P1 generation, we will have the two parents, here round seed, pure breed round seeded plants, pure breed wrinkle seeded plants. Then you have the gametes. Gametes, normally we use a circle to show the gametes. So from both plants we only get one type of gametes. Here it is capital R, here it is simple R. And when they combine, what do we get? We get the F1 generation. F1 generation. F1 generation. So now we need to write the F1 generation. We have the two gametes here. When both the gametes combine, we only get one type of plant. So to show that, we only need one square. We only need a square to show the generation. Here it's going to be capital R, simple R, but all the seeds are going to be round seeds. Then what do we get? From this, we get the gametes and then we get the F2 generation. We can show all those together in one box. Instead of again showing the gametes in circles, we can directly show the gametes in the Punnett square itself. So if we do that, here you will have a large square where you need to divide this like that, because we will have the gametes here and after that we will have the F2 generation. Now in the previous one, you all can remember, students, I showed the gametes in circles, capital R, or there it was the tall and short character, capital T, simple T, capital T, simple T, and then I drew the Punnett square. But you can omit that particular step because again here also we are showing the gametes. I told you all I wrote it with different colors there, but I am not going to do that here. We have the gametes. What are the gametes? Capital R, simple R. Here also we have the gametes, capital R, simple R. Then you show the formation of F2 generation. Here R and R combines, capital R, capital R. You know it's a round seed. Then capital R, simple R. It will be a wrinkled seed. Again, simple R, capital R, but always remember you write the capital letter first. Capital R, simple R, that is also round seed. So, R, capital R, or capital R, simple R, all are round seeds. So, 3 round seeds. And you have 1 simple R combination. You will get 1 wrinkled seed. So, from this, again, you can write the ratio. We have already written the ratio. Round is to wrinkled, 3 is to 1. And the other ratio, RR, capital R is to capital R, simple R is to simple R. That is the same ratio. 1 is to 2 is to 1. So, I told you all, the character that is expressed is known as the phenotype. So, the phenotype ratio is going to be 3 is to 1, and the genotype, the factors shown here are going to be the genes. They are the genes. So, that is the genotype ratio, that will be 1 is to 2 is to 1. So, before I have discussed the gene concept itself, I have introduced these terms to you so that you become familiar with those, and when we discuss the gene concept, you will understand better. So, I am sure you all can understand this clearly. So, if I quickly go back, you can see the Punnett square method. Parental generation, here we don't have to put P1, parent generation. So, that is capital R and simple R. Then gametes, R and R. F1 generation, round seeds but capital R, simple R. Then you have the gametes and F2 generation, both written inside a Punnett square. Capital R, simple R, gametes, capital R, simple R, gametes, 2 parents. Then here you have RR, capital R, capital R, simple R, and capital R, simple R, and simple R, simple R. So, from that you all know round seeds and wrinkled seeds. Is that okay, student? So, that is for round is to wrinkled, 3 is to 1. The observation of Mendel's experiment. Is that clear to you? So, if this is clear to you all, now I'll move on to the next slide where we will try to understand how these ratios are correct according to probability, or how we can use probability to explain these ratios. Probability of getting an outcome. So, here if we say outcome, getting a round seed or getting a wrinkle seeded plant, or getting a tall plant, getting a short plant. So, those are all outcomes. So, after this, students, I will move on to the next slide where we look at an assignment. So, assignment. Select another pair of contrasting feature of pea plant using 20.1 table. That is the table we used before. State pure breeding dominant character or pure breeding recessive character using symbols. Build up a Punnett square to show the inheritance of this monohybrid cross. So, since it is a name of the scientist, it's always better if we write it in Punnett square in capital letters. So, we need to show the inheritance of this monohybrid character. So, we can do that. We don't have to do the symbol method. We only have to use the Punnett square method. So, if I go back to the table, here you can see, students, we have already used round and wrinkle seeds. Then we have used tall and short character. You can use any of these colors. I will use the color of the pod, green and yellow. Green is the dominant character. All F1 generation is green, and yellow is the recessive character. So, you all know for the symbol, we will be using capital G for green color, simple G for recessive yellow color. So, that is what I am going to use, color of the pod. So, here. So, we have selected the character, color of the pod. State pure breeding dominant character, pure breeding dominant character, that is green. So if you use symbols capital G, capital, then we need to write the pure breeding recessive character, that is yellow color. Here we use simple G. Dominant character green, recessive character yellow. Dominant character capital G, recessive character simple G. Since it is pure breeding dominant character, you have to show both the factors. Pure breed plant, it has two factors. Then what do? We build up a Punnett square to show the inheritance of this monohybrid character. So what do we have first? To write the Punnett square, we need the parent generation. Parent generation, you need two squares. G, capital G, simple G. So those are the parents. From that, what do we get? We get the gametes. Gametes are indicated in circles. So we have one circle here, another circle here, because capital G and simple G from the first parent and the other parent. Then what do we have? We have the F1 generation. So here we need to have the F1 generation, combination of the two gametes. So there we will have the first generation, only one combination, capital G, simple G. So you know the color of the pod will be green color. Then from that we get the gametes and the F2 generation. We can do that together. So what do we do? Gametes and also F2 generation. So there, you need to have a larger box with smaller boxes there. So here we need to divide it into 3 rows and 3 columns. Then you have the gametes in these two and these two boxes. Capital G, simple G, capital G, simple G. Then when they combine, capital G, green color pod. Capital G, simple G, again green. Capital G, simple G, you know always the capital letter has to come first. All 3 are green color pods. Then simple G, you have the yellow color pods. So from this, you all can see green is to yellow is equal to 3:1, and also capital G is to capital G, simple G is to simple G. The ratio is, there is 1 here is to 2 of these there, and so here 1 simple G, 1 is to 2 is to 1. So those are the ratios that we get from for the second generation, and that is how we build up a Punnett square. So this is for your assignment. We selected the color of pod, pure breeding dominant character. green, and pure breeding recessive character yellow. So capital G for dominant character, simple G for recessive character. From that we take the parent generation in boxes, then gametes in circles. You get the F1 generation, only one type. Here also if you want you can write all green pods. And then you take the gametes and the F2 generation. There you show the gametes on the inside those boxes and the F2 generation here. So we get 3 green color pods and only 1 yellow color pod. So that is the observation, and that is how we build up the Punnett square. So far students, I have done 3 examples. Tall character, short character, then round seeds, wrinkle seeds, and now the color of pod, green and yellow. I'm sure you all have a good understanding of this. You can even try with the other characteristics also. Purple and white flower, axial and terminal flowers, inflated and constricted pods, any of the other characteristics. So then you will have a very good understanding about how to write and explain the occurrence of F1 and F2 generations using symbols and also the Punnett square. So with that, I'll move on to the next slide. So this is the explanation of patterns of inheritance using probability. So this is something students that you all are familiar with. You all know what probability is. Now when there are more than one outcomes for a particular activity, say for example you toss a coin. Now the coin has a head and a tail, 2 faces. So the possible outcomes are the occurrence of a head or a tail. So then there is the probability of the occurrence of the outcomes. Probability of getting a head, probability of getting a tail. So that is where we discuss probability. And we can explain the pattern of inheritance using probability. You got the ratios. Now you can remember occurrence of tall plants is to short plants. 3:1 ratio. But if you look at the combination of factors, capital T, capital T, capital T, simple T is to simple T, simple T. That ratio was 1:2:1. So these 2 ratios can be explained using probability. We will see how we can do that. So when any 2 different objects combine randomly, the result will take place according to a particular pattern. So that is the occurrence of different outcomes. As an example, consider random combination of X and Y objects. So if we consider the combination of X and Y, now you have X plus Y and you have another X plus Y. Now this X can combine with X can combine with Y. At the same time, Y can combine with X, Y can combine with Y. So there, what will you get? You can get X, you can get XY. Again, you can have YX, and you can have YY. Now in this is XX. Now here you have XY, YX. So both 2 different occurrences, 2 different outcomes of the same occurrence. So we can take these 2 as XY and this is YY. So again you can see a similarity with the inheritance. XX, XY, YY. So if you have dominant and recessive characters similar to capital T, capital T, capital T, simple T and simple T, simple T. So this is how the occurrence happens. So 2 characters present in parents inherit into offspring randomly. Carry out below activity to identify the probability. Probability means the ability of an incident to happen. So here, if we say probability, probability of getting XX, there are 4 different outcomes. Out of those 4, we have 1 chance of getting, now 1, 2, 3, 4 possible outcomes. Out of that, 1 chance that we will get both X. Here you can see 2 chances that we can get a combination of X and Y. So here we have 2 chances. And here we have Y and Y, that is again 1 chance. So these are the probabilities. We have a probability of 1/4 to get X and X. Probability of 1/4 to get Y and Y. And probability of 2/4 of getting 2 different outcomes, X and Y. So this is something related to probability. I am sure you all already know. Now we will do the activity, or we will actually try to understand the activity what is given to us in the next slide. So here we have an activity. I'll first read the activity, then I'll explain to you all. So identification of probability patterns obtained by a bead experiment. Divide the class into 4 to 5 groups. Provide 2 vessels with mixture of 50 beads, white beads W, and 50 red beads, capital R, to each group. Instead of beads, can use buttons or seeds with 2 different color. Can be used. Here there is a repetition, students. This has to be. So instead of beads, can use buttons or seeds with 2 different colors. So either that way, or you all can understand the meaning. We need to have 2 different colors. The same object, 2 different colors. So you have to make sure if it's the bead, the same size, and buttons also similar. If you had different shapes, then of course when you draw out the buttons, there will be a difference. They have to be of the same shape. So that is required, 2 different colors, so then you can identify them easily. Take out a bead from 2 vessels at a time and note the color of 2 beads with tally marks in the relevant column and return them into the same vessel. So you all know what tally marks are. To denote the outcome, we use a line, and then after 4 outcomes, the 5th one, we cross it. You are familiar with tally marks. Then likewise, continue it for 50 times and tabulate results. Present the results to the class. So this is what we are going to do. Each group will have 2 vessels. You are going to have 2 vessels. Now in both vessels, you will have 2 colors. You will have 50 white beads and you will have 50 red beads. So here you will see now 50 W white beads and 50 R red beads. Here also you will have 50 W white beads and 50 R red beads. So both vessels contain a total of 100 beads, 50 white and 50 red. And simultaneously you are going to take out 2 beads. And then you note down the colors and you have to put the beads back to the vessels. If you don't put it back, you won't have the same number for each occurrence. Always you need to have 100 in this and 100 in that. So for that, you have to put the bead back to the vessel. So when you take out the beads, there are so many different possibilities. So you can have a white bead taken from the first vessel, and the second vessel also it can be a white bead. Or else you can get a white bead from this one, or else you can get a white bead from that one. At the same time, you can get a red bead here and red bead there. These are the possible outcomes. I will draw that and explain it to you. So here we can have both red beads. Now you can see white, 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 red, red, white, red, red. So these are the possible outcomes. Now we are drawing beads from two vessels. Both can be white beads. This one can be white and this one can be red. This can be red and the second one can be white. Both can be red beads. So two beads are taken at a time and there are different combinations. Now isn't this similar to what we discussed before? Occurrence with X and Y. So here also you have the observations. Both can be white. One is white, the other one red, red, white and red, red. So four different outcomes for the same activity. Four possible outcomes. Is that clear to you? So we do it 50 times and we mark it tally marks. So you know how to put tally marks. You put 1, 2, 3, 4, then you cross it. So that is 5 times it has occurred. Like that we put tally marks. So that is how the activity should be done. I'm sure you can understand that. So then I'll move on to the next slide where we have a set of data and from that we can just try to understand what happens there. So results. The first group, now here you can see red. You can see 5, 10, 14. So 14 times both the beads are red. Then here you can see 10, 12 times red and white. 5 and 3, 8 times white and red. And here you can see 15, 16 times white and white. So that is a total of 50. That is a total of 50. Like this, the second group, like that if you have 4 or 5 groups, you have so many observations and you add them together. And you all know always when there is an experiment activity carried out related to probability, the more number of times you repeat, the observation, the result will become more and more accurate. So here 14 and 16 somewhat close to 50. Whereas 12 and 8 again somewhat close to 9, 10. So like that when you repeat it more, the ratio will be more better. But using this also we can calculate. Now we will see what happens. Fill the above table using results obtained by the above practical using tally marks. Then you calculate times of red beads and probability of getting red beads. So how do you calculate that? Here you have red. So what we do is total is 50. Out of that you have obtained 14 times red. Then red white 12 out of 50. White red 8 out of 50 and white 16 out of 50. Then you can simplify this. Now 14 out of 50 you can take it as 7 out of 25 or we can have it this way itself. Like I said, if we do it many times, if we repeat it more, if we take the total, this will come to a more closer ratio that will give you a better idea. So we will see if we get that ratio what we can do. So here times of red beads and probability of getting red. Times of red white and probability of getting red white. Times of white red and probability of getting white red. Then times of white and probability of getting white beads, and using those probability values find out the ratio of red white red and white. So if you have a lot of observations, you will get a probability where you get red as 1 over 4, red-white as 1 over 4, white-red as 1 over 4, and white-white also 1 over 4. All the probabilities will be equal, and that will be equal to 1 over 4. That is after you add up all the observations of all the groups and finally calculate the probability. Here I have only done it with the values obtained by the first group. But when it is 5 groups, you get a better observation. Is that clear to you? So you will get a probability ratio like this. Then I'll move on to the next slide. From first and second vessels, the probability of getting red. So that is what I wrote before, 1 over 4. The probability of getting white, that is also 1 over 4. The probability of getting red, 1 over 4. And the probability of getting white, 1 over 4. But in these you can see, both are red color, both are white color. 1 over 4, 1 over 4. But the third and fourth instances, red and white, white and red, getting 2 different outcomes. So we can combine that together. Now getting red and white or white and red. So to get the probability of that, what we do is we multiply 1 over 4 for red and white, 1 over 4 for white and red. We need to add them together because 2 different colors together. That will give you 2 over 4, that is half. So like this we can calculate the ratio. Then you will see 1 over 4, both these together is 2 over 4 and this is 1 over 4. So 1 is to 2 is to 1. A ratio similar to what we obtained from the Mendel's experiment. I will explain this further again in the next slide as well. So here, in third and fourth instances, both show the same incidence. Both vessels, the probability of getting red, that is equal to 1 over 4. The probability of getting white, that is also 1 over 4. But probability of getting red and white, that is 1 over 4 plus 1 over 4. That is 2 over 4. The ratio between probability, you will get it as 1 is to 2 is to 1. Red, 1. Red, white, 2. And white, 1. So the ratio is 1 is to 2 is to 1. According to Mendel's monohybrid cross, the ratio of F2 genotypes, capital T is to capital T simple t is to simple t is also 1 is to 2 is to 1. So you can understand the relationship. Similarly, the probability ratio of bead experiment compatible with the genotypes of F2 generation. So here you can see this is the bead experiment. You draw out 2 beads at a time and then you get the results. You calculate the probability. You get the ratio. The ratio is 1 is to 2 is to 1. Same as the ratio obtained for the F2 generation of Mendel's experiment. So then, now that you all understand how probability works and how you can explain the gene concept using probability. So, 4 different outcomes, but out of that, 2 are different. Capital T, simple t, only 1 outcome each. But getting capital T and simple t, you have 2 probability, 2 out of 4. Therefore, we get the ratio as 1 is to 2 is to 1. So, that is something you all can understand. With that, students, I will move on to the next slide. So this is a do you know slide. The probability of getting the head or tail when tossing a coin is half. That is something you all know. So you get either head or tail and the probability is half. Why? Only two outcomes. So you either get a head or a tail. So half. The bead experiment is similar to relationships of probabilities using two coins at a time. So like I told you all before, you can toss two coins at a time. You can get both heads, both tails or head and tail combination. So to find out head head, head tail, tail head and tail tail probabilities, the probabilities obtained from each coin has to be multiplied. So then the probability is half into half. Why do we multiply here? For one head, the probability is half. The other head probability is half. So if you want both to be heads, it will be half into half. If it is one coin, only two outcomes. If there are two coins tossed simultaneously, there will be four outcomes. So to get the probability, you multiply half into half. That is 1/4. When the same incident takes place in two ways, example, getting head tail and tail head, like we had before, is the total of separately taken probabilities. So you get head and tail, 1/4, tail and head, that is also 1/4. So you add them together, you get the probability as half. This is same as getting white and red beads, red and white beads, both combinations. Or having capital T, simple T, or capital R, simple R, or capital G, simple G. When we did the Mendel's experiment, we tried to illustrate, explain the occurrence of F2 generation. We got the same ratio, 1:2:1. The same thing is explained with tossing coins as well. So the previous activity we discussed taking out beads, drawing out beads. Here we have tossing of coins. Either way, you will get the same ratio, 1:2:1. That is what you all have to understand clearly, students. So with that, I have come to the end of this chapter where we explained or we used symbols to explain the observation achieved by Mendel when he did the experiments. He got the observations, he got the ratio, that is the phenotype ratio 3:1 and genotype ratio 1:2:1. So we were able to explain that using the symbols as well as Punnett square. I'm sure you all have a good understanding of that. So with this, I'm going to end this chapter, and in the next chapter, we will discuss the gene concept and how we can use genes to explain the same inheritance that was observed by Mendel and other related information as well.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.